hope you can see my slides. Yes, thank you so much. And I also would like to start by just greeting everyone. Good afternoon, good evening, or good morning from where you are. I'm Ali Penyangu. I work with the PHRC as a program coordinator. So I just want to give you a small background of why we are even talking about the strengthening regional antimicrobial resistance research systems in Africa, and that will probably give more context to why we are meeting today and why we are just having this conversation today. Yeah, if I would start with some background, is that today we are meeting because we have this partnership that was concluded between the Swedish International Development and Cooperation Agency and the African Population and Health Research Center in 2021 for jointly implementing the programming initiative on antimicrobial resistance program. So through this partnership, we have been able to run or to collaborate with 22 partner institutions across 13 African countries, and we have cohorts going back to 2020, and we have a cohort going 2021, 2022, and recently a cohort of 4 partner institutions implementing also AMR research project in their respective countries. So yeah, this partnership has given us an understanding that we need to have a regional system approach to strengthen antimicrobial research across Africa, just for 2 big reasons. Why is the high and growing AMR burden in Africa? Also, we, from the experience and interacting with the JPIAMR project, we have seen that there are a lot of happening across Africa. We have many project funded projects doing some research or some other interventions on AMR across Africa, but they remain vertical projects and they tend to be silo working in the silos. So there is no. So from that experience, we see that there is a need to have to connect these projects and bring together or everything that we are learning from the work that is being done by different projects, and we can turn the experiences we have learned from JPIAMR project into a regional learning and action platform. So I will just briefly talk about some of these 4 elements that pushed us to think about these regional efforts to strengthen AMR research across Africa. So yeah, one of the, as I said, that's number one. It's because of the burden of AMR across Africa. So you know that it has been estimated that Africa carries the heaviest global burden of AMR, and that one it is explained by the weak surveillance system that we have across Africa, with the also high use of unregulated antimicrobial use, and we also have seen the limited lab capacities to detect and start taking care or controlling antimicrobial resistance. So all these efforts, so we think that we should do something to stop the rise, or to control the rise of AMR, because if nothing is done, that could still undermine the country's efforts to universal health coverage and health security and also sustainable development. As I said, also we have seen various or several efforts, several projects implementing research on AMR, and but most of them they are just project grant-based bound. So where we know this experience of grants, when you implement projects, when the grants over, other efforts stop from there, and no one is focusing on the next steps, and that's really something that we think with these efforts that we are starting today can provide some solution to that, because we should not be focusing on just implementing research grants on the stop from there. We should also think about how to connect with others and keep pursuing the impact of the project. So we think that this vertical project, if we start working together, we can bring together, fragment the evidence and have something impact across the regions. That's number 2. justification the reason why we need these regional efforts to towards AMR research across Africa. And as I said, we also have we want to leverage on what we have learned from our experience from the JPIAMR fund management initiative with our fund at CEDA. And because I briefly talked about it, with this JPIAMR funding with the support from CEDA, we were able to we have been able to implement 22 collaborate with 22 partner institutions across Africa, and we have already some evidence from over 19 publications done, and we have had visits with these partner institutions, and a lot of things related to capacity strengthening has happened with students, and so we think that there is already some good basis for us to start and connect these projects and strengthen local AMR systems across Africa. Yes, this is just the reason why we think we should be thinking about the regions is that you look at these countries that we have been able to collaborate with, and over 13 countries across Africa, you see that it's not yet the entire Africa, but I'm sure there are other non-SEDA funded JPIAMR program that could come in, and we come together, we share experiences, we share findings, we share evidence collected, and we can have something tangible as we move forward. So we are suggesting this webinar series to create a regional learning. Not as I said, most of these projects have closed out, but we think we should not stop from there, because evidence generated should be used in informed decisions that should inform decisions or efforts that should guide the prevention and the control of AMR rise across Africa. And we also, that's our thinking, that also is aligned with APHRC's vision, that our vision or our mission is to transform lives in Africa through research. And we believe in just, we do generate evidence, we strengthen research and data capacities in Africa and research and development ecosystem. But we also think after that, we should not stop from there. We need to engage policymakers to engage in making decisions that impact lives. And how that should be done, it's what we are starting today. And we need this evidence to be discussed, and we need to engage with people to communicate evidence that we have collected from this project, so that people can start embracing the evidence and put them to good use and come up with the solutions to controlling and preventing AMR. Yeah. Now, these webinar series that we are proposed to start today, the goal is to strengthen country systems in cross-country partnership for AMR research and surveillance and response across Africa. And we hope to facilitate knowledge exchange as we are starting today, because project, as I said, project have some evidence generated, and we think that it's time now to start exchanging this knowledge, not just publications. As I said, a lot of publications have been made, but it's good to also comment to have the comments of the people whose experience have been much related to this publication. And we also strengthen regional cross-sectional networks among researchers, because, as I said, having been funded through grants, we should not stop from the grant cycles. We should go beyond that and implement the project to impact the lives across Africa. Yeah, we are proposing to act as a starting. We have identified from the mapping of the project evidence. We have seen that we can start with 5 thematic work streams. We have the first thematic work stream is AMR surveillance, infrastructure, and innovation. The other one is the one we are focusing on today is the environmental AMR monitoring and control. And we also have time to talk about the economic and the behavioral strategies for stewardship. And we also have a work stream on antimicrobial animal health and antimicrobial use monitoring systems. And there will also be the opportunity to focus on One Health governance and policy systems. So, yeah, we think that this webinar series will be, we hope to carry out them for the next 5 months, starting from today. And we we hope that will be a good opportunity to learn about what is happening on AMR research across Africa. Yeah, I think I would like also to express our gratitude to SIDA for having funded all these projects, and we hope that we also continue to work with them as we strengthen the capacities of African institutions, African countries in deep primary research. I think I can stop from here, and over to you, Anne.
Thank you, Alipio. I'm sure some of our participants might have some questions or comments for you, but I will ask that we note them down and save them for the Q&A session. However, as we progress, you can utilize the Q&A feature, and our speakers will be able to respond to the questions as the webinar continues. Now, I would like to welcome our speakers, both professors, Charles and Sidiba, and maybe just to give a highlight. In the interest of time, I won't read their impressive and long bios, but I'll be able to share this along with the webinar resources. Our first presentation will be from Charles Masembe. He's from the University of Makerere, where he's a veterinarian and professor of molecular genetics and evolution. And he has a lot of teaching and research experience. I'm sure he will tell us more about his work during his presentation. He'll present on pairwise. Allow me to use the abbreviation, but he will expound on what is pairwise. After that, we'll have Professor Sidiba from the Chemical and Environmental Engineering at the National School of Engineers. Here, he's an associate professor and the inaugural head of the Department of Biomedical at the Institute in Mali. And Professor will also tell us about himself and his work. Prof. Charles, the floor is yours. We are looking forward to hear about PRAs and the things that the project has done during its implementation. Welcome.
Okay, thank you. Thank you very much, Anne. And happy to see all of you. Many of you have turned up to this webinar, and we don't take that for granted. So we are very, very happy that you are here. Innocent, Gucci, Gizat, Alvin, Aaron, and Grace, to mention but a few. All of you are very welcome. Please, somebody tell me if you are seeing my slides.
Yes, we are, Prof.
Yes, we are.
Okay, fine. And thanks for that introduction. And you mentioned that I should tell us what pairwise is. This is an acronym for dispersal of antibiotic resistance and the antibiotics in water ecosystems, and the influence on livestock and the aquatic wildlife. We chose this because many times when we talk about AMR, it is more pronounced in the human side, but then it leaves out the environment, it leaves out the wildlife, it leaves out the livestock. Yet we are in this together. Again, this week we are commemorating the World AMR Awareness Week, and the theme for this AMR Awareness Week is act now and we protect our present, or protect what we have now. Because if we have protected it, then we know that the future will be bright, not only for us, but also for our children and their children. What was the overall goal of this pairwise project? One, we wanted to find out the dispersal of 3 agents, mainly the antibiotic, the antimicrobial resistant bacteria, and then the genes that are responsible for that in different environments, in wastewater treatment plants and in animal places, by comparing the 2 aspects, climatic regions and also seasons. So we did that in the wastewater management to see the wastewater management practices, the water bodies, and in Sweden, in Europe, but also in Africa. Again, contrasting environments. In Europe, we had Sweden, Spain, Norway, and in Africa, we had the Uganda and the Tunisia. So it's just a map to show the countries where we're operating, Uganda, Tunisia, and others. So again, now concentrating on Uganda, we looked at two contrasting environments. We had one in Western Uganda and another one about 600 kilometers away, that is in northern Uganda, in the Gulu region. Gulu region has got less anthropogenic pressure, and the Western Uganda has got quite a lot of anthropogenic pressure. These are pictures which we got during our work, and it is not uncommon when you find a river like this one, and then there's quite a lot of stuff which has been washed on the banks with the domestic use and the bottles and the medications, vials of drugs that have been used, even those which have not been used. A few meters away, it is not uncommon to find the human speaking water for a domestic use, of course. And now this comes with the risks. You ask yourself, what is the risk that this can cause antimicrobial spread of the resistance? Again, in the same river, I would say again, almost at the same spot, except at different times, the animals have been brought here to water. And it is a common practice that people in this community, they always bring animals and they drink water from there. So you ask yourself, are these animals being exposed to water which is contaminated with the antimicrobial resistant bacteria, antimicrobial resistant genes, and what is the likelihood of this spreading in different when we visited the farmers, we asked the farmers, hey, do you use any antibiotics on your farm? So no, we don't use. But if you walk around the farm, you will quickly see evidence that actually the farmers have been using these medications by themselves. We have got birds which migrate from Europe during winter all the way up to Uganda and even beyond. They move along the Nile. They move along the Nile, and some of them rest in particular spots in Uganda. Birds of interest to us were the gray-headed gulls and the Egyptian geese. But we also picked interest in our domestic, birds in Uganda. For example, the marabou stork, it is common in many human dwellings, and then the pied crows, because then they live in common places where humans are, maybe abattoirs where there's a landfill and all that. So we ask ourselves, what is the contribution of these birds? Do they play any role in AMR? And then we had to wait patiently and collect droppings. For example, these are droppings of one bird, marabou stork, and this is one of our students when we are out there collecting samples. You go to the landfill and you find many marabou storks, and you still go in and collect the droppings, dung, and you take swabs and you go to the lab to check ARB, ARG, and the antibiotics. So this is just a schematic flow of the work that we did, which I could take that if you are attending this, you have an interest in AMR, and we have done this. Maybe it could have variations from wherever you are. But we did all this, the water we collected and we checked for residues, and the dung and the droppings we also collected and checked for, went through this procedure up to sequencing. And yeah, we did some of that work. Those are some of the pictures which we did showing what we were doing. Now, our colleagues also in Europe were doing this work, and in Sweden and Spain, they had very, very low levels of ARB, ARG in wastewater treatments, but treatment plants far away. But if you come to Uganda, The results of antibiotics ASTs show total resistance among the most commonly used beta-lactams, amoxicillin, all the way up to erythromycin. And this was greater in Western Uganda. The effluent samples from the hospital, the wastewater treatment plant, also registered the presence of ESBL, ESBL producing E. coli and K. pneumoniae isolates. We had 20 E. coli and 10 K. pneumoniae isolates, which carried the CTXM, blood CTXM gene, and 22 carried the blood TEM gene, and 8 carried the SHV gene, which makes us get worried as a country. In Sweden, only 1%. They tested thousands of samples, only 1%. We'll see what we found in Uganda. And they did not have any differences in the seasons. The same happened in Spain. Still, there were a very low percentage, 2.7, and then 0.3 for the carbapenemes. So that is really not so worrying for them. But we'll know why. We'll have an idea why, because then they are very vigilant. And then they didn't have any differences in the different herds. So Spain, Sweden, they were okay, I should say. Now in Uganda, the cow parts showed presence of AMR. Cow parts is cow dung. The blood CTXM gene and the TEM gene and the SHV gene were amplified in quite a number of samples. And this was quite a lot in the cattle farms, in the river Ruiz catchment, as well as in the river Aswa. So when we go to the droppings of the birds, the marabou and the pied crows, we pick those samples from abattoirs, landfills, and land sites. The CTXM gene was amplified in 37 out of 38 E. coli and K. pneumoniae isolates. We also identified the blood TEM gene in quite a number of samples, and the blood SH gene also was amplified. So you see, everything here was the CTXM, the TEM, the SG, SHV were all positive and in high numbers, and not only at isolates, but also at amplification and sequencing. Now, a sneak peek look into what we got from the CTXM genes. I'll just use the CTXM gene as an example for E. coli and K. pneumoniae. You remember the map which I showed you, it had this Western Uganda and Northern Uganda. So the CTXM gene had two large clades, clade 1 and clade 2. But now when you look at clade 1, it exists in these areas. But now if you see this thin line here, this area is in Gulu, is in northern part of, this one is southern Coronorian is southern part of Uganda, and Gulu is northern part of Uganda. So you see this here, it is mixed. It does not differentiate between the north and the western area, meaning that the haplotype, this haplotype here is shared between the different regions in the country. But now this haplotype clade 2 is restricted to Gulu only. Now, when you look at the CTXM from K. pneumoniae, it also existed in two haplotypes or two clades. One clade in this area here and another clade in this area here. The unique thing here is that this haplotype, even if it existed twice, it did not exist in Western Uganda. So the question here is, if you're instituting a control mechanism, a prevention mechanism, a diagnostic mechanism, if you do AST and say, okay, the CTXM gene is present here. If you go an extra mile, you may even find more and more information there. Now, what are the implications for this, what we have found out? I imagine that each country that you are coming from to attend this webinar, you have got an AMR strategic plan. And for us here in Uganda, maybe in East Africa, would like to, the plan says, okay, we have to strengthen the knowledge and we have to use this evidence to contribute to revising and forming policy. And also we have to invest quite a lot in research and use those innovations to inform a policy and implementation practice. Question is, when are we going to, this information, how can we use it to inform our policy, to revise our control mechanisms, to make informed decision, so that the decisions that we make are not informed by passion, but instead they are formed by reason. We have been able to, as a pairwise, as team pairwise, this surveillance mechanism, this surveillance approach, this research approach has not been used before. So we have provided novel surveillance results and tools which can be used to form mitigation strategies to prevent dissemination of these antibiotic-resistant bacteria in different environments. And this can also be used in areas where you suspect there's AMR for you to make investigations. And then at a global level, we feel we have contributed to the antimicrobial resistance in One Health approach. And this is in support, this is in agreement with the larger international bodies, the World Organization for Animal Health, the FAO, and also APHRC aspiration, and the Africa that we want, all speaking to Agenda 2063. This is not my work alone. I was the PI for the Uganda section, the co-PI for the international, for the project. But then everyone in the pairwise team is acknowledged. APHRC JPIAMR is acknowledged. And also my employer who gave me the time and the platform to do this work. I thank you.
Thank you, Charles, and keeping time. I'm sure that the participants have questions, and please use the chat feature or note down for the Q&A session. I would like to move to Professor Sidi. Welcome. Please let us know what nature means. It's not the nature word that we know, and we are looking forward to hear more about the project. Welcome.
All right. Thank you so very much, Anne, and hello, everyone. I will share my presentation. Is it in presentation mode for everyone?
Yes, now it's in presentation.
Nice, now.
Okay. All right. So my name is Sidiba, Associate Professor at the department, a newly funded Department of Biomedical, Chemical, and Environmental Engineering at the National School of Engineers, which acronym you see, NIABT, it's French. So be aware of the difference between the English and the French. All right. So our project, as you see, is called Nature, which stand for nature-based solutions to reduce antibiotics and pathogens and antimicrobial resistance in aquatic ecosystems. So just to give you which are the members of this project, the consortium is made of 6 partner institutions in 5 countries, 4 in Europe and 1 in Africa. I was the only African represented in our school was the only African represented in the consortium. And the consortium was led by CSIC in Spain, Victor Matamoros, and he was a very good leader throughout the implementation of the project. So, and then we have a CIMAR, and CSIC in Spain and CIMAR in Portugal are 2 research institutions. And then you have the National School of Engineers in Bamako, which is, of course, a higher education institution. And then you have Aarhus University in Denmark. And then one of the very interesting partnership we have in this consortium was we have a private company called Kilian Water, and we will see shortly why this company was on board. And then we have the Karlsruhe Institute of Technology in Germany that was also part of the consortium. And then we have the website of the project is still updated. You can still go there, and as you see, it's on the top of your screen, and learn from what was executed and what are the ongoing project, because our project is still ongoing, although it ended about a year ago. So the main goal of the nature project was to provide scientific evidence about the use of nature-based solution to reduce aquatic pollutants. And the three aquatic pollutants we were focusing on are the antibiotics, the AMR, and some of the resistant pathogens at the river catchment scale. So this is what you see these pictures. As you can see, we focused on tertiary wastewater treatment, secondary wastewater treatment, and also we have the estuarine area, and so some natural wetland, meaning a flowing river. So the specific goal was objective was to evaluate first, can NBS reduce pollutant in wastewater treatment and in river catchment? That was one of the main objectives of our project. And then we were also monitoring the pollutant transformation byproducts in the NBS, meaning the nature-based solutions, and identify the diagnostic indicators for future monitoring assessment of NBS. This was part of the project that the objective that was executed mainly in Mali, because we were trying to make so to establish statistical relationships between some of the water quality, general water quality parameters, and then the reduction of those three pollutant we were focusing on, and to see what are the relationship when some of the general water quality parameter that everyone can measure, when those changes, how that affects the one of the three emerging contaminant, we can call them. And then to assess the impact of NBS implementation on environmental and human health, that was mainly executed in Spain and Germany. So when we talk here about nature-based solutions, as you know, that's a very broad concept nowadays. But in our case, we were talking about natural and constructed wetlands. And as you see on these pictures, the first one on the left of your screen, we have a vertical flow constructed wetlands. We have aerated constructed wetland, and then we have bioelectrochemical constructed wetland. So when you talk about constructed wetland, those who are in the area know that there are mainly three variants of the constructed wetland. You have the vertical flow, the horizontal flow, and then the free surface flow of constructed wetland. But with clean water, the private company based in Denmark that I talked to you about, we brought them on board because this company is developing, making a very innovative constructed wetland, modifying the three main technologies of this constructed wetland that are known. That's how he came up with the aerated constructed wetland and then the bioelectrochemical constructed wetland. So you can learn about this if you Google simply Kilian Water. You can find all the work he is doing throughout Europe. It's one of the leading companies implementing this technology in Europe nowadays, since more countries are going toward using nature-based solutions to treat effluent. And then you have also, these systems are used to treat secondary wastewater treatment, tertiary wastewater treatment. And then when you go to Denmark, Spain, and Portugal, there are areas in the estuary where you have the natural wetlands. So, and those natural wetlands are within the area of rivers flowing sometime and just in the nature like this. And then in some places in the estuary, that's the case in Portugal. So we went and monitored all the antibiotics and the AMRs and some of the pathogens that we could find in the water there, and then to try to see what are the attenuation effectiveness of these systems in the contaminants. So just some finding that we were able to find in these systems. For instance, in a tertiary, that was I'm just showing an example because it's too much result that we cannot show here. So there was a tertiary wastewater treatment plant in Spain where we monitored 30 antibiotics, and then we try to see what are the AMRs that are linked to these antibiotics, and also what are the most virulent pathogens that are also linked to these antibiotics. So you see here, out of the 30 monitored antibiotics, so 8 was found in the wastewater treatment plant from Spain. It's near Barcelona. And then now we also, that some of that wastewater was also treated in some of the systems. You see the horizontal flow constructed wetland, the surface flow constructed wetland, and we made a comparison. So what you see here on your screen on the right side, you see that from the tertiary wastewater treatment plant in Barcelona, so some of these antibiotics that you see ciprofloxacin, ofloxacin, so all these different antibiotics that of the 8 that were already found in the wastewater treatment plant, you see that there is only 46% on average attenuation from the tertiary wastewater treatment plant. But if you take that same wastewater with the same concentration of these antibiotics, you see that in the constructed, whether it's the horizontal flow or the vertical flow constructed wetland, you see that the attenuation removal goes up to between 71 and 77%. So it means that basically the nature-based solution system perform better than the conventional tertiary wastewater treatment plant. That was a takeaway from this particular investigation. And then we did in Mali. In Mali, we didn't have a constructed wetland that we worked on yet, but it was to investigate the presence, because this study is the first of its kind in Mali, in our knowledge, at least in the literature. So we investigated effluent coming out of the 5 main hospitals in Bamako. If you know Bamako, Bamako is a city that is across the Niger River, which is the main river in West Africa. And then these are the 5 hospitals that you see here on your screen on the left side. And so much of that effluent is flowing all the way to the river through the sewer system. And sometimes even the sewer system are not very good maintained, as you can expect. That happens very often in Africa. So what we found out is that we have 12 antibiotics that we have found in the wastewater coming out of these 5 hospitals. And these are the antibiotics that are the most consumed and the most prescribed as well. But the most important information we found, if you go to Europe, when they do their investigation, the concentration of these antibiotics are in the level of nanogram per liter. But here we found them up to from 10 to 38 microgram per liter. It means it's more than 1,000 times the concentration that you have in Europe. Of course, that can be explained by the fact that these antibiotic, the wastewater treatment plant that you have in Europe, you don't have them in Africa. That's the case in our hospitals. In our hospitals, we have some of the sludge system, but that's not efficient at all in removing antibiotic. So it means we are spreading the antibiotic into the environment, going up to the Niger River, which is also the drinking water source of the city, and the drinking water source of many of the cities that this river run across in West Africa, all the way from Guinea all the way to Nigeria. So you see, you have 9 countries. So many of the cities across the river use that river as source of drinking water system. Since even when you take drinking water treatment plant, we know that the conventional treatment plant do not completely remove some of these antibiotic residues in the surface water. So that means it's a concern. And on top of that, these antibiotics are the one who are generating the resistance in the environment. So it means that there is a problem to address it. And here, these are just the concentration in the graph that you see here. It's just the concentration of the ciprofloxacin that I am highlighting here, where you have the concentration up to 38 microgram per liter. So now the question that one can ask is, does the constructed wetland affect the AMR composition? So, well, we have samples from all the sites, meaning Mali, Spain, Portugal, that we shipped to Germany. And then they found out that, yes, some of the AMR indicators genes can be reduced sometime in 1 to 3 log-fold log unit reduction. So it means all the way you have 99 or 99.9% reduction of the AMR composition when you have it in the wastewater and it is treated through the constructed wetlands. So now, for our this was the scientific part of the project, but the project goes beyond just the science. You know, it was a very good collaboration, coordination, mobility, and synergetic actions. And we were lucky that this consortium had all the expertise within it that we needed: analytical chemistry, microbiology, chemical and environmental engineering, biology. So, and then we also managed to have students and some of the researchers working in the lab mobility in between those partner institutions. And then we also have an impact and knowledge output. We know that these constructed wetlands, especially when they are modified, they can be tailored to remove some of the contaminants. So we believe that this would have a big impact in contributing to the implementation of the EU water framework and urban wastewater treatment directives, but also on the SDGs and One Health approach, both in EU member countries and in Mali. So we were also able to put together some platform in social media with our website. We talked about some teach brochures and some promotional videos that you can access to see all the work that we have generated through this project. And then we also engaged at least 36 stakeholders across these 5 countries, both in Europe and in Africa. And you see in Mali, for instance, we had 8 stakeholders that we were engaged with, and those are involved sometime in the One Health approach within the country. So that was a very good and important. And then as far as impact and knowledge output, we were able to generate 3 publications. We were the leading author of the 2 publications, and then we were also contributed to one of the review paper that our consortium has published. And then other key achievements include 1 PhD fully completed with the support of the Nature project. We have also 1 master student who completed his master on the project. And we have another PhD candidate who was partially supported through this project. And then I mentioned the 3 peer review papers. We have 1 conference posters that we were able to also present at one of the APHRC workshop that we were fortunate to attend. And then we have 2 international research stay in Europe. One of my PhD student was in Spain. Another master student was in Denmark to learn about the constructed wetland with the clean water implemented in some of the municipalities in Denmark. And then we have built some laboratory capacity with the acquisition of key analytical and experimental equipment. And there is an ongoing follow-up activities and demonstration of the Nature project here in Mali. Hopefully I will get a chance to talk about that in the questions. So take-home messages: antibiotic and microbial resistant genes have been found in wastewater, reclaimed water, river stream, but also in estuaries areas in Europe and Mali. The constructed wetland as nature-based solution can effectively remove antibiotic and ARGs from wastewater, and they can also be effectively applied in river streams and estuaries. And in our case, we are using this system to improve the water quality of the Niger River here in Bamako. There is an ongoing work. And then we also have contributed to build capacity on AMRs in Africa through the JPIAMR nature project. And lasting impact is ongoing, of course, as I mentioned earlier. So thank you very much. I would like to take this opportunity to thank APHRC for this opportunity, and of course CIDA that funded our nature project. So I would like to say, as you know, nature-based solutions are living solutions, we like to say. So the nature project is still a living project. You can go to our website. It is updated regularly because our collaboration between these institutions is still ongoing beyond the project. And I would like to thank all the partners. You see them on your screen. And thank you very much for attending this session. And we will be happy to take any of your questions. Thank you very much. Over to you, Anne.
Wow, thank you, Sidi. I hope the participants have enjoyed both of the presentations as much as I have. Please let's show our appreciation to both Charles and Sidi for the wonderful walkthroughs of their projects, which I consider successful, and I believe you do as well. And maybe just to follow up and to dig a bit deeper into what you've talked about, the research projects you've walked us through, you were selected competitively. And perhaps it would be good for participants to learn from you what lessons you learned throughout the implementation period. What support did you receive, and how helpful was this support or help in terms of project management to be able to achieve the impact that you have? And perhaps you can also add in when you are carrying out the work on the different research on AMR in your countries, what surprised you the most that in your years of experience you thought, wow, that is new, or I didn't think it was at this large scale? I think we can begin with Charles.
Yeah. Thank you very much. And thanks, Sidi, for a wonderful presentation as well, and the audience for being very attentive. I see the numbers are increasing by the minute. When you are looking for funding to do this kind of research, it's not easy, but it's not impossible. Perseverance are very important, because when we applied for this JPIAMR, this was the second time when we were applying, when we went through. The first time we applied, we didn't go through. And then, so when we applied the second time, yes, we were successful. So if we had not persevered, then we wouldn't have reached this far. We had to trust each other. I mean, you're collaborating with people in Europe. You don't see them very often. So you have to trust each other. They have to trust you and believe in you. And the trust goes with confidence. So we must have confidence and trust in each other. And the mutual respect. Mutual respect is very important, especially when you're doing North-South collaboration. Mutual respect is very, very important. And then what I also saw here is that research is moving very fast, but painfully, government is not moving very fast. But we do the research for scientific purposes, but with the aim of contributing to government's activities to provide what it should provide. For example, I mentioned the policy. The results that we've got, they should inform policy. But is it my responsibility as a researcher or as an academician to begin revising policy? No. So through APHRC, through CIDA, through many agencies, we must narrow that gap so that researchers and the government, they sit on the same table. Government usually sees researchers as, oh, you're going to find fault with what I'm doing. No. That's not the aim. Good things can be improved. So the research can inform policy for better lives. That's why you saw that in Europe, researchers and government work together. So you saw the percentages are very minuscule. What surprised me as a PI, as a co-PI, I've told you I'm a veterinarian, molecular geneticist. Now, our colleagues in the human health side, they were not very conversant with the tools that we use in molecular genetics. Now, when we show them the tools that we use in molecular genetics, then they say, oh, we have only been seeing the tip of the iceberg. The problem is bigger than what we thought. So that really surprised me. I will stop at that.
Thank you, Charles. Interesting. Sidi, what about you?
Yeah, thanks so very much. And I have to confess that I agree on whatever Charles has just said. This is true for me too. So what I would like to add is, sometimes as a researcher, you have to be open to offer that comes to you. That was my case. My contact with this consortium was through Jochen in Germany. I had some work with him previously, and he's the one who contacted and said, oh, we want to do this. We think you are the right person for our consortium coming from Africa. We need someone from the global side, South. I said, oh, okay, why not? Of course, I like to work on this. So that's how the whole thing started. What I learned from this is it is very important when you build a consortium to make sure that you have within the consortium the expertise you needed to execute the project you are proposing. In our case, for instance, we didn't need to try to have, during the implementation of the project, any external partner or outsourcing of some of the work. You see, everything went, very seamlessly across the partners. That was something I really enjoyed during our project implementation. So the thing that surprised me the most, I have to say, there are two things. One is, who expected COVID-19 outbreak during we just started our project and it was COVID-19 outbreak and all the shutdown and everything. So things as simple as a chemical reagent, don't even mention some of the equipment we needed on our site here to execute some of the project, it was impossible to get. So how do we go around that? So, well, we relied on our partners. So it means communication, how that played a critical role was important. So anything that we could not analyze here in Mali, when we mention, so someone would say, oh, we can do that in Spain. Oh, send us a sample. We can do that in Germany. Guess what happened? Then you have the Ukraine war started. And then samples get stuck in airport for 1 month because everything is being screened. So you have to be prepared for challenges that would always arise during project execution and engage with your partner to see how together as a team you can find a solution to move forward, because you cannot stop your project because because of these challenges. That was of the most stressful side. And then the thing that I enjoyed the most during that was the good surprise. Researchers are always small communities, especially when it comes to Africa. When I was discussing my project with some of the colleagues and other researchers on other aspects of the One Health approach, they were so impressed that we are going to use a nature-based solution, because they said, oh, this infrastructure can easily be put together in Mali without much investment. So it means there is a way to treat the effluent coming out of the hospitals without polluting much the Niger River using that approach. So that was something that I was very pleased about. Thank you.
Thank you. Evidently, partnership is very important, and partners that are on the same page and you're working together, not competitively, but you're on the same goal. And it's good to hear that one of the reasons that has made this project successful is because of the partnerships and how you've worked together. And Charles, you talked about funding and the application twice, and I'm sure you've had other projects beyond Pairwise, and maybe just to highlight, what are some of the challenges that come with funding for research projects in AMR and beyond, and particularly for projects or countries in Africa, why do you think we have these challenges and how can we overcome them? If you can give us some insight.
The biggest funder is government. So we have to lobby. Our domestic challenges for health and everything, we say they are global, but they start domestic. If a country does not have a healthy population, then whom will it collect money from? Who will be the workforce for this? So we must lobby government. We must break the box. We must get out of the box and we think openly. But now that you ask about that, I also bring in something which I've realized. We all know there's a recent call about a JPM from a new version of JPIAMR. And you saw the problems that we have. In Africa, AMR is 23%, what. In Spain, in Denmark, in Sweden, 1%, and you saw the figures. And there's no African country which is qualified to be part of that application. So where is the problem? The biggest health problem is here in Africa, but we are not funding our research. But now Europe is giving a lot of money to address that 1%. Maybe our bosses should talk to Europe, should talk to their financiers. We should talk to inside and see how we can break the network. And that would go to, maybe I would say, if I was given more money, I would say, okay, now look, instead of working in isolation, and we have got all these good results from CDBA's group, my group, and other groups across it, put them together. What can we break? Where can we break? Such that we have found, okay, there's AMR in cows, there's AMR in birds, there's AMR in this. How can we break that cycle? Instead of next time somebody coming and say, okay, there was AMR in marabou stock. Now I want to find if there's AMR in the ducks, if there's AMR in the bird, in the weaver birds. So I think we have to put up a concerted effort and lobby for funding aggressively, convince our politicians that if they don't have a health population, then they have no voters. Thank you.
No voters indeed. Sidi, research
Yes.
projects, I believe that all JPIAMR funded initiatives, one of the aspects was to build local capacity, or rather to strengthen it and pave way for sustainability. To what extent do you think you've achieved this within the Nature Project?
Thanks very much for this question, Anne. Yeah, I would say that actually, when we got this grant, of course, with the support from SIDA on my side, I was just starting my lab because I moved from one institute to another, and I was starting my lab. The first equipment, as basic as the lab bench in my laboratory, I got it from the Nature Project. And then we were able to get all the other equipment that we needed to buy to conduct general water quality analysis. So basically, I would say this Nature Project was really very, very helpful in starting up my lab. Capacity building in terms of materials and equipment. That's one thing. The other thing is, I mentioned in my presentation, one of my PhD students was fully covered to earn his PhD on this grant. And then another master student also earned his master's degree on this Nature Project. And then I have another female, the two were male, and then another female PhD student who were partially funded through the Nature Project. And the grant, my portion of the grants was relatively very small compared to other grants that I've been involved with. But it was very, very effective in generating capacity building in terms of training students, I would say the next researchers, and laboratory strengthening. So that's the capacity, and it goes beyond that, because I have the master student who was on the project is now pursuing a PhD on the same project idea of the nature. And then I have two other PhD students who were also looking at some of the nature-based solutions because of the nature project to take some of those aspects into their PhD work. So I would say, yeah, the capacity building was really very important. And then beyond the capacity building in terms also of laboratory and then human capacity, there was the raising awareness, because sometimes capacity is not, it's sometimes immaterial, right? When you go across our school today, our school is core engineering school. So things like AMRs and, pathogens, those are not things people are used to talk about in our school before I came there. But through this nature project, when you talk about AMR in our school, everyone knows about it. Everyone knows what are the consequences and what should be done in terms of policy change, so that the spread of the antibiotic and AMRs could be controlled. And this is all thanks to the CEDA grant. Thank you.
Wow, thank you. We are continually amazed as we hear the wonderful things that you are able to do. And to move on to our last questions to both of you, we've talked about sustainability impact, and for sure, the message is clear that these projects were there to lay the foundation for, long-term impact, even as other initiatives or projects come on board. But just to say you got the opportunity for additional funding to continue the project or to build on it, what other research questions or areas would you like to explore? And maybe you can start with Charles and then finish with Sidi.
Okay. Thank you very much. I've alluded to this, but for emphasis, we have found out where these AMR, where these genes, how they behave, where they are located. Now, the next question is, how do we solve the problem? How do we reduce the AMR? How do we reduce the ARB? How do we reduce the ARG from the environment? How did they go there in the first place? How do we stop them from going there? So if I was given funding, I would be addressing that. And maybe from this other side, I would approach aspects of the humans themselves, human behavior, and how they behave when they are getting medications with their animals, the professionals, the paraprofessionals, all those entities which come across medicines. They need to be educated. They need to be mindset change such that they cannot. You don't have to police them. In Europe, when I've visited there several times, I say, "Oh, I've got a headache." Now, my host, Carl, usually say, "Okay, before you take a Panadol, first take 2 glasses of water." I say, "No, back home I would just be taking a Panadol. No, first take water." So you see, it is ingrained in them. So how do we cause a change in here in Africa? So to address reduction, removal, mindset change, yeah, since the evidence is already there, that's where I would do channel my efforts. Yeah, thank you.
Thank you. Sidi?
Yeah, thank you very much. This is, I consider the nature projects when you consider Mali side, we established a baseline study. And since we are very concerned about the Niger River, if we were lucky to get funding on this project idea, is to expand this study, because as I said, the Niger River is huge. Even in Mali, if you go to the Mali, the big country, as you know, if you go to the central part or in the northern part, we know there are issues there related to the spread of antibiotic within the river, and then what are the bugs that you can find that could impact the health system in terms of infectious diseases. This is something to be addressed first, and then we are hoping also to collaborate more with other African institutions within the Niger River Basin. That's one thing. The second thing we would be, since we in the one of the most important aspects of the nature project was it was not just investigation and then no, it was also the solution. We are using constructed wetland to address the spread of AMRs in the environment. So we are already with one of the PhD students who was a master student on the project. He is building in our school now a pilot system of constructed wetland to see how he can optimize it. At the end, we would like to take this into a larger scale, partnering maybe with one of the hospital to see how we complement the pre-treatment system that we have in place in the hospital. Because if you consider the constructed wetland, it's a vegetated system, and it's you can even use ornamental plant. Can you imagine in a hospital where you have people sick who sometimes need to go out and take fresh air, and we can walk around these systems and then have even fresher air coming out of the system while the system is also removing some of these contaminants. This is really something that we are looking into to take it at a larger scale and also to expand the study. So yeah, we wish that SIDA could continue this grant, but I agree with Charles. You know, we cannot always rely on Europe to fund our research. Sometimes we have to make our politicians, our decision makers, to contribute to the research agenda in our countries. Because if this was not an agenda that was not addressed in Europe, maybe we would never have a nature project in Mali, you see? But now that it is done, we are still pitching people locally here so that, this whole area of AMRs as part of the One Health approach could be considered, because sometimes the environmental aspect in the One Health approach is neglected in our countries. I talk from what I know. I'm talking about something I know. Even the vet part is sometimes neglected, but the environmental part is even more neglected. So we are trying to pitch the decision makers in the countries, and this is one of the workshops I am organizing very soon, because as I said, our project is not ended fully yet in terms of the sustainability. So we will be invited all the stakeholders so that the people be aware of the need to address this issue with local resources. Of course, if we can have SIDA on board again, it would be fine. Thank you.
Cindy, both of you are lucky. Marcus from the Swedish International Development Cooperation Agency, SIDA, is here. And he might know someone who knows someone, or he might be someone who can do something about the additional funding. Marcus, you might, I'm sure our participants would love to hear a few remarks from you, and welcome. We're so pleased to have you in this session.
Thank you very much, Anne, and all presenters. It's a great pleasure to be with you today, because it's very exciting. I mean, that APHRC started this webinar series, and I would like to thank you and colleagues for organizing this webinar series and gathering so many people. If I saw it rightly, I mean, it was more than 100 participants covering all over from all over the African continent and beyond. So that is really a great success. And I would also thank you, APHRC, for all the work that you did and that you are doing together with us, with SIDA, but then mainly, of course, with the projects and the project PIs and participants. I mean, this became so much more than just putting funding into some research projects, because you were really actively involved in building capacity on the ground together with the research team. So that was and is still very much appreciated from SIDA. And then I would like to congratulate you, Charles and Sidi, for winning these very competitive grants. And I mean, the presentations were great, and it's great to see all the important results that you and your teams have achieved during the years. And I think during your presentations and now in the discussion, I mean, you said it all. I mean, there's so many components, so many bits and pieces that are important. I mean, it's of course you need to have a lab and you need to have the staff capacity. I mean, that the research can be done, and then you have hopefully research results that you can publish. But then, I mean, they need to get out of the, I mean, you can call it ivory tower or whatever, and there needs to be an impact. The results need to be used to make a change. I mean, lead to policy, need to change in procedures, behavior, and that is of course an important part of the work, and that is not always easy to achieve. And then also you touched upon the challenges with the One Health approach. I mean, that it's still very much that you work in silos, that the environmental and the veterinary part is not as much recognized as the human health part of the One Health sphere. So that is also something that I think you brilliantly addressed, and that we have to continue to work with. And then the capacity building and the sustainability. I mean, it was our hope from the beginning. I mean, we know it was funding, but it was not an awful lot of funding. But it seems, I mean, it not only contributed to you setting up relations to other researchers in Europe and maybe also in other regions. And of course, getting research results, but it contributed to building capacity, everything from having a lab bench and the equipment and the reagents that you need. But then great to hear, I mean, that students have been involved in the projects, have been educated, they have graduated, and they are hopefully continuing this type of work. And then I see it, I mean, in the chat here, I mean, the people are really interested, and they're asking for networking opportunities, for opportunities to work together, and that is of course also part of the capacity building and building sustainability. I mean, creating interest and getting people working together. And of course, the funding aspect is an important one, and it is a little bit, or it's very unfortunate that our possibilities are limited at the moment. I mean, to continue funding these projects, but there will hopefully be more opportunities in the future. And you mentioned it. I mean, the importance of national and also regional funding. I mean, talking to your national governments and other regional bodies and organizations is probably very important. I mean, to secure funding for this important type of work and addressing the AMR challenges in the future. And as you know, I mean, people always or quite often talk about AMR as the silent pandemic, and I hope your work and this type of events, this webinar, contributes to, I mean, making people, the researchers, but also populations, policymakers, decision makers aware of what the AMR challenge is, what it implies, what the consequences are. I mean, to making it not being a silent pandemic, but an issue that people are aware of and also want to address. For example, with funding, then. So I'm very, very impressed about what you all have achieved, and I'm really proud. I mean, that Sida was a part of that, a little part of that journey. That is that we're in the middle of. It's not the end of the journey, and I'm looking forward to follow the projects and the upcoming seminars, and I hope we will stay connected in the future as well. Thank you very much.
Thank you so much,
Marcus.
Marcus.
Thank you very much for taking time to attend.
Always great to hear your insights and for the support you've provided to APHRC and to the project. We cannot appreciate you enough. Okay, I think it's time to hear something from our participants. I know we have some questions in the Q&A feature, and I invite Alipio, perhaps even there's a question for Marcus there, Charles and CD to look at that. But then I can ask that if you have a question and you'd like to speak, please raise your hand and I will unmute you so that you can either give your comment or raise your questions to the speakers. we have about 15 minutes to go, and I plan to not steal anybody's time. So please raise your hand. I see Adugna, your hand is up. Can I allow you to talk? Okay. He's taking time. So let me unmute David. Please raise your hand if you'd like to say something. Okay. David, you can unmute. Okay.
Yes, thank you. Thank you for this good presentation that you have had. I would just want to ask, is there an AMR network, regional network that has been formed or is yet to be formed where we can have expertise, what do you call it, expertise details that you can use when you're coming up with joint or multidisciplinary, studies or proposal presentation, development? That's aspect number one. The aspect number two, I would just want to comment on something that has just been raised up there. It is being said that you're working on silos. That's not the issue. You're not working on silos. The issue here is that we have decided that the funding organizations, you see, they have remained at the corporate level. What do I mean by that? At the corporate level is that they want to give out funds to specific research organizations, but these funds, in one way or the other, they don't trickle down to the people down there who needs these funds for the aspect of research. So that has been a very, very, bigger challenge that I think that needs to be addressed and something like that. I don't want to take more time. Those are the two key issues that I wanted to probably to raise. Thank you.
Thanks, David. Who would want to address some of those issues raised and questions by David? Alipio, I know you have something to say about the network of future plans of.
Yeah. Yes, thank you, David. I think we would not. I would not say that the platform is not yet there, because what we are trying to see is if there is something happening already, we should concentrate our efforts and ensure that we have the network working on that area that we were mentioning. But as far as I know, I try to see, as I told you, there are many initiatives going on AMR research across Africa, but you just alluded to it. It's as if because most of the people they are grants based, the ongoing initiatives on AMR, they are grants based. So most of the time the grants, they are just pursuing objectives and some deliverables, some timelines. So after the grants, nothing happens, and the people lose track of what they have done, and evidence is lost out in many other things. Although evidence is lost in the shelves. So yeah, that's what we are trying to push for the project that we funded by CEDA, and we hope that we can even bring together many other people who would like to continue to share the evidence and build the network as we said. Yes. Also, from what I know is that from the different projects funded by JPIAMR, they collect some data, and there is a repository of data which you can access if you want to explore environment. You can access. I think you would need to consult their website to be informed about the process to get the data and use them. Yeah, for sure there is data that's being collected, and you should be able to get organized and access it and use it, and so yeah. The other comment that you were saying about the funding organization, I don't blame them, because what they are giving is the opportunity. So no one has prevented people from the regions of to come together and use the evidence they have generated and collaborate and network and move forward. They have something bigger. Like now, let's assume that you've had the findings presented by Charles and the CD. So you can see there is a possibility for networking and collaborating and have some joint response. So this is the kind of thing, the reflection that we would have to continue, or we would like to see happening, so that people we learn from the experiences, what takes in Mali, and can easily be replicated in Uganda to solve the issue of ARGs observed in different ways. So that would be my contribution, but my colleagues also can come in and supplement what I've just said. Thank you, and over.
Thanks, Alipio. CD Charles, anything to add?
The regional AMR, yes, there are some networks there, but maybe we need a herdsman, and that herdsman might be APHRC. I mean, we converge on the subject AMR, but then after we have done our bit, we stop. We need to sustain the effort. I see we need to sustain effort such that we always together, newsletters, communications, seminars, conferences, funding, exchanges, all those kind of things. And the funding remaining at corporate level, I think, David, you're right, but being frank, it is what it is. The funders have to reach somewhere, and then we have to do the last mile. The government and us, the researchers, who are the frontline actors, we need to do the last mile. Yeah.
Thank you, Charles. Indeed, we do have to do the last mile. CD?
I think they responded, Charles and Alipio. What I just want to say is, talking about a network of AMR expertise in Africa. Actually, one of my student, a PhD student, wrote a very nice review paper on AMR in aquatic systems. I was going to propose to Alipio because she did an investigation on the publication that she could find everywhere on the issue in Africa. So I think a network is worth if there is a collaboration, something ongoing. So I would like to propose that those who are interested in the AMR in the environmental side, especially the aquatic systems, to be in touch with Alipio. I will be sharing that manuscript. And Charles, by the way, I will be very pleased if you can come on board on this manuscript so that we do a review of the work that has been done. She already done a very good work on the manuscript. And then we can give an African perspective on how to even address the issue before the decision makers, whether it is on the African organizations or even the national I don't like to use politics, but sometimes those are the people who make the decision. So that could come to the paper, and then I think that could even be something that would lead to more grant. At least I will be reaching out to Alipio to share the manuscript at the stage it is, and to propose anyone who can come on board, and then we will give a perspective on AMR. And this is not going to be a paper from my student only, it's going to be a Pan-African paper. That's something I would like to put up.
Fantastic. Great to hear that. So I don't see any questions, but I know questions have been raised and addressed in the Q&A session, and we just have 5 minutes to go. So I'll call on to one more person to raise their hand, but if they don't do that in 1, 2, 3, then maybe I can bring the session to a close. Fantastic presentations and engagements. We have learned a lot. I myself have learned a lot, and the engagement from the participants in the chat says a lot as well. To our participants, please expect the presentations and the recording, and we'll have a short form evaluation of some sorts where you can tell us what you pick from this session and how we can improve, since, as Alipio mentioned, we are planning, this is a webinar series, so we are planning to have 4 more sessions that we run up to next year. So if there's nothing else, thank you so much, Charles. Thank you, CD. Thank you, Alipio and Marcus. Thank you also, David, for raising the questions, and all our participants. We've really enjoyed the session. The 1.5 hours has gone by very fast, and I wish you a good evening until the next session. Goodbye.
Thank you very much, APHA. You did a great job. And
Thank you.
Goodbye.
Thanks to
Thank you very
everyone.
much.
Yeah.
thank you, Marcus. Take care. I leave you.
Bye-bye.
Charles,
Take care.
Thank you. Bye.
Thanks,
Bye.
everybody.
Bye. Thank you.
Bye.
Thank you. Bye.